Polychaete nereis is a species of predatory worm that can feed on dead organic matter or filter feed. They are found in estuaries in the northwest Atlantic as well as the Mediterranean Sea, the Black Sea, and the Caspian Sea. They come in a variety of colors with two dark bands along the length of their bodies. Here we see several different species of Polychaete, but for this particular study, researchers were interested in Polychaete nereis. Nereis influences sediment in estuaries because they burrow and disturb the sediment in a process called bioturbation, which is crucial to estuarial ecosystems. An estuary is where a river flows into an ocean and fresh water and salt water mix. These ecosystems are very delicate, but when in balance, they are very productive. The goal of the research was to study the effects of the density of the Polychaete nereis on benthic flux in 4 different estuaries. Benthic flux is the rate of movement of particles between the sediment and the water at the bottom of a body of water in an area known as the benthic zone. Researchers sampled sediment cores from estuaries in Italy, Sweden, Portugal, and England. At each of the 4 estuaries, sediment cores were collected, as were native species of the Polychaete nereis. Nereis were added to the cores in the abundance found in the estuary of their origin, leading to relatively high and very high densities, while control cores did not receive worms. After nereis worms were added, the sediment cores were allowed to sit overnight in water. Researchers then measured the flow rates of nutrients like oxygen, ammonium, nitrates, and phosphorus between the sediment and the water, and the rate of denitrification. Denitrification is a step in the nitrogen cycle that converts nitrates to nitrogen gas through a process called reduction, which is then released into the atmosphere. Overall, sediment cores that contain nereis worms had an increased flow rate of oxygen, ammonium, and nitrates between the sediment core and water column. There was more oxygen and more ammonium released from the sediment in the water when nereis worms were present, and there was more nitrate uptake by the soil occupied by nereis worms. Adding nereis worms to the cores also increased the rate of water denitrification, meaning that more nitrates were converted to nitrogen gas in sediment cores that contain nereis. Adding nereis worms to the cores had different effects on the flow rate of phosphorus depending on the location of the estuary. Adding nereis worms to the cores from the Italian, Portuguese, and English estuaries had a significant impact on the flow rate of phosphorus, but adding nereis to the core from the Swedish estuaries had no impact on the flow rate of phosphorus. The Italian estuary had the highest flow rate of phosphorus from the sediment to the water. In the Portuguese estuary, phosphorus moved from the water to the sediment in cores without nereis worms, but when nereis was added, phosphorus movement changed direction and moved from the sediment to the surrounding water. In sediment cores from the English estuary, cores without nereis released phosphorus from the sediment, while cores with nereis took up phosphorus into the sediment. Overall, nereis have a significant impact on the flow rate of nutrients in estuary environments due to their ability to disturb sediment at the bottom of estuaries when digging tunnels. However, the specific effects of nereis differ depending on their environment. The Polychaete nereis is essential to sediment ecology and health. Oxygen that is dissolved in water is essential to the diverse organisms that live in aquatic environments. When there is a lack of oxygen in an aquatic environment, such as in dead zones following algal blooms due to excess fertilizer use, the animals that normally live there either die or move to a more hospitable location. In the natural habitat of nereis, the benthic zone, there are naturally occurring low oxygen levels, making it especially important to return oxygen from the soil to the water, where it can be used by other organisms.